This PowerPoint will cover the microscopic anatomy and physiology of skeletal muscle. Skeletal muscles are organs of locomotion. Most of them are attached to bone, and the length of a muscle is measured between its attachment points. Skeletal muscle tissue cells are extremely long. In fact, in an individual who is 6 foot tall, the sartorius muscle could have a cell in it as long as 2 feet. They are so long that they're often referred to as fibers. These muscle cells are striated, and the striation means that you can see horizontal lines going through the muscle cell, and they are actually caused by the alignment of the myofilaments that are within the muscle cell. Muscle cells are cylindrical, and they're multinucleated. They need to be multinucleated because they are so long. They need to have more than one DNA structure maintaining the muscle cell itself. And they are under voluntary control. The structural organization of skeletal muscle begins with the muscle fiber. An individual muscle fiber has a connective tissue wrapped around it, and these individual muscle fibers are then grouped together in bundles called fascicles. Then that fascicle will have some connective tissue wrapped around it, and then bundles of fascicles are wrapped around by an exterior connective tissue, which forms the whole muscle. Muscle cells have a microscopic anatomy that you can see on the slide here, that it's made up of a sarcomere, myofibrils, myofilaments, known as actin and myosin, which we'll discuss in detail in subsequent slides. So you heard me mention in the other slide about the different connective tissues. Epimysium is the connective tissue that covers the entire whole muscle on the exterior portion. Epi is on top of, so it's on top of and wraps completely around it. The perimysium is the connective tissue that wraps around the individual fascicles or bundles of groups of muscle fibers. And then endomysium is the connective tissue that wraps around the individual single muscle cell fiber. All of these three connective tissues will blend together to form the tendons that connect muscle to bone. And as I mentioned in the previous slide, we would talk about the microscopic anatomy of a skeletal muscle cell. Skeletal muscle cells have a lot of the basic same anatomy as any other cell in the body, but because they are so long and they are contractile tissue, some of their structures have unique names. For example, the sarcolemma is the plasma membrane that wraps around the entire muscle cell. It has unique structures called T-tubules that extend off of the sarcolemma and go actually into the interior of the cell, and they wrap around these protein structures called myofibrils. They are also found in between an organelle called the sarcoplasmic reticulum. The sarcoplasm of a muscle cell contains cytosol and organelles like any other cell in the body. And in the cytosol, you're going to find ions and glucose and amino acids, enzymes and glycogen, myoglobin. I don't have it on this list. And it has all these things that it needs, and it's like a soup. It has all the stuff it needs for energy and to facilitate a muscle cell contraction, which is its job. It also has organelles that are like other structures. It has the mitochondria, and then it has the myoglobin, which is an interesting organelle that's pretty unique to muscle cell. That's kind of what makes it red. And it stores oxygen in it because that is the preferred method of ATP production is cellular respiration. So it needs to have quite a bit of oxygen stored up. The sarcoplasmic reticulum is the structure I mentioned before that is the T-tubules from the sarcolemma runs in between those, and it creates what's called a triad. And the sarcoplasmic reticulum's main function is to store calcium. And when it stores the calcium, when the T-tubules are depolarized, they'll open voltage-gated calcium channels that are on the sarcoplasmic reticulum, releasing calcium into the cytosol or in cytoplasm. Myofibrils and sarcomeres. Myofibrils are proteins that run the entire length of the muscle fiber, and they are segmented by some structural units called sarcomeres. The sarcomere itself is the smallest contractile unit of a muscle fiber, and its length runs from Z line to Z line, as you can see in the diagram on the right. Within the sarcomere, it contains myofilaments called actin and myosin. The myosin myofilament is thick, and it has these heads protruding off of it on opposite sides, and it's typically portrayed as red in most diagrams. The actin is a thin filament, and it's a double helix, and it's portrayed in blue in the diagram on the right. And it has binding sites called myosin binding sites that the myosin heads will bind with when it is stimulated to facilitate a contraction. Tropomyosin and troponin. Tropomyosin is a linear protein that covers the myosin binding sites. You see it in kind of a darker mustard colored yellow going across the double helix of the actin, as you see in the diagram. The troponin is a three-part protein that holds or locks the tropomyosin in place. It unlocks when it binds with calcium. So when the calcium that is stored in the sarcoplasmic reticulum is released by depolarization of the T tubules, it'll bind with that troponin. That troponin will unlock and pull the tropomyosin away, and those myosin sites will be exposed, allowing the myosin head to attach to it and begin the contraction process. The sarcomere myofilament organization. As I mentioned earlier, it's the arrangement of these myofilaments that are responsible for the striations that are visible on a histology slide. Now what we have here is a diagram representing a sarcomere. And you see we've got a Z line to Z line. But each zone has a specific name, and it's because of specific things that are contained in it. For example, the A zone or A line, it contains both myosin and actin filaments. The H line or H zone has only myosin in it. And then the M line, which it doesn't show an M line here, but I have black circles denoting it, it is the center of the sarcomere, and it has just the myosin filaments in it. The Z line contains just the borders, the edge that hold the myofilaments attaches to them. And the actin is attached to the Z line via another protein called actinin, which not only attaches it to the Z line, but it'll attach it to the actin myofilament in the subsequent sarcomere. Then there's another little protein, a coil protein called tintin, and it's represented in yellow here. And it attaches the myosin filaments to the Z line, but it'll also then attach to a actin filament in the subsequent sarcomere. So you see there's always an alternate pattern of the sarcomere. The I zone that's on the sarcomere contains just the actin myofilament. So I believe we've covered all the zones. The I zone is just actin, the A zone is actin and myosin, and the H zone is just myosin. Desmin and dystrophin. These are proteins that attach the actin to the sarcolemma. The main one I'm really focusing on is dystrophin, and that's because that is the protein that is involved with a condition called muscular dystrophy, particularly Duchenne's and Becker's muscular dystrophy. Children with Duchenne's muscular dystrophy do not produce any normal dystrophin. And because they don't have normal dystrophin, every time their muscle cells contract, it makes a little tear in the sarcolemma that eventually damages the muscle cell completely, and the protein gets replaced with fat, and it really becomes no longer functional. Children with Becker's dystrophin, they can make some dystrophin that functions, but not much. So these children will also have some clinical signs of muscle weakness with their condition. Both muscular dystrophies, Becker's and dystrophin, only affect males. I'm not going to read all of this to you. This is the information that I talked about the different lines on the sarcomeres, the different bands, and about the desmin and dystrophin. The sliding filament theory is that during the contraction, actin and myosin filaments slide across each other to shorten the sarcomere, so that nothing actually shortens or gets smaller during the contraction. As you can see in the top one, when the muscle cell is in the relaxed state, you have that space, that H line, that is only myosin. Then when a contraction is facilitated and the actin is pulled towards the midline by the myosin, you see the crossover. And you can even see a clear difference in the black and white image of a histology slide of how it changes those lines. And so it's just the sliding of these filaments that actually shorten the sarcomere. I want to point out that a muscle cell does not shorten all the way across from attachment to attachment. It just shortens at each sarcomere, which ultimately shortens the entire muscle cell. A skeletal muscle contraction begins with a stimulus from the lower motor neuron. The cell body originates in the ventral horn, and the neuron fiber exits through the motor nerve root, and it'll bind with the whole spinal nerve just proximal to the intervertebral foramen, and then it'll reach its target skeletal muscle cell via a peripheral nerve. Motor neurons have several telodendria. We already just talked about that in our nervous tissue module. Each ending of a telodendra will have a synaptic bulb at it, or a axon terminal, or a synaptic knob. It has so many AKAs. And these axon terminals, synaptic bulbs, will form the neuromuscular junction at the muscle belly of a muscle cell fiber. So when it's at the muscle belly, that's usually the center of the whole muscle. So the neuromuscular junction, when it causes depolarization, that wave will spread out from the NMJ towards the attachment point. So it's like a pebble in a pond. It'll initiate at the neuromuscular junction and then spread out to each end of the muscle. The axon terminal of a motor neuron comes in close proximity to the sarcolemma at a neuromuscular junction. They don't touch. They have a space in between called the synaptic cleft. These neuromuscular junctions are also surrounded by Schwann cells to insulate it from surrounding fluids and to help facilitate the depolarization wave that arrives at the axon terminal. Some of the structures that we need to remember at the neuromuscular junction is, of course, the axon terminal of the motor neuron, the presynaptic membrane, which is on the axon terminal. The postsynaptic membrane at a neuromuscular junction is the sarcolemma, but it is referred to as the motor end plate when it's at the neuromuscular junction. And it is convoluted, and these convolutions are also referred to as the subneural clefts. I mentioned before there's a space in between the axon terminal and the motor end plate at a neuromuscular junction because the axon terminal comes in close proximity. It doesn't actually touch the sarcolemma. So there is a synaptic cleft, which can be anywhere from 20 to 30 nanometers or 200 to 300 angstroms. In this diagram, it's showing that vesicles releasing neurotransmitters, which in skeletal muscle is always going to be acetylcholine. It shows those green dots of acetylcholine floating across the synaptic cleft because they move by kinetic energy only. And it shows them binding to a ligand-gated ion channel on the synaptic cleft. But it also points out that these ion channels need to have one acetylcholine neurotransmitter molecule on each side to open it up. Just think like of using two hands to slide open a elevator door. That is mechanically, if you will. So it needs to have two acetylcholine molecules to open a ligand-gated ion channel on the sarcolemma. The resting membrane potential of a skeletal muscle fiber is negative 80 to negative 90 millivolts. That's a little bit higher than it was for a neuron. If you remember, a neuron was negative 70 millivolts. However, the threshold is the same as a neuron, negative 55 millivolts. Typically, each action potential at a neuromuscular junction is strong enough to facilitate a muscle cell contraction. This action potential typically causes a membrane potential of 50 to 75 millivolts. If you look at the diagram, you'll see that the peak of an action potential in a skeletal muscle is about the same as it was in a neuron, positive 30 millivolts. This strong action potential arriving at a neuromuscular junction has 3 times more end plate potential than is actually needed to initiate the contraction. This provides a high safety zone against neural fatigue, meaning that almost every time you send a voluntary signal down a peripheral neuron, motor neuron, to a skeletal muscle junction, that's going to stimulate that muscle fiber so that it always works. If it doesn't, then there's some kind of pathology going on. However, if you're constantly, if there's so much repetitive stimulation, it can cause the muscle cell to fatigue, typically from running out of oxygen stores to make ATP, or you could have just depleted the neurons' acetylcholine. This does not take very long for the acetylcholine to regenerate, and just taking a few breaths of air will replenish the oxygen, and that muscle cell can actually respond to a stimulus again. So here are the events at a neuromuscular junction. A lot of this is going to be reviewed because the first part of it is very similar of what happens at a junction between two neurons. So what happens first? The action potential reaches the axon terminal. This action potential will cause calcium channels to open. It only needs to be about 4 to 11 millivolts. These calcium channels, when they open, will cause the vesicles to migrate to the presynaptic membrane and release their neurotransmitter acetylcholine into the synaptic cleft. The acetylcholine will diffuse across the synaptic cleft via kinetic energy and will bind to nicotinic receptors, because all acetylcholine stimulatory receptors are nicotinic, and all nicotinic receptors are ionotropic, and we want these receptors to open to allow ions in. And so they're also referred to as ligand-gated ion channels. These receptors will require 2 acetylcholine molecules to open them up, and once they open the ion channels, sodium will flood in, and that's because the concentration gradient of low sodium inside and high sodium outside of the cell is maintained by this sodium-potassium pump. A tiny bit of potassium will exit, but not so much because of the rapid influx of sodium, and will kind of push potassium out of the way. This will cause the sarcolemma to depolarize at the motor end plate. And once the motor end plate depolarizes, the action potential will spread in both directions, because I mentioned before that a neuromuscular junction is typically at the muscle belly, and it'll go in both directions towards the insertion points. This is also referred to as propagation of the action potential. And just like in neurons, an action potential, once it is started in a skeletal muscle, it is an all or nothing event. It will continue in both directions, and the muscle fiber will contract. Immediately after the acetylcholine has been released, within about 5 milliseconds, it's going to be removed from the synaptic cleft. Some of it is broken down by acetylcholinesterase, and is broken down into choline and acetate, and the choline is taken up by the axon terminal to be recycled and used as a precursor or a element to create more neurotransmitters. Some of it will just float out of the synaptic cleft via diffusion. The removal of acetylcholine will cause the ion channels to close, and then there won't be any more sodium flooding in, and depolarization will have timed out. And this prevents continued stimulation of the muscle fiber. So there are medications that actually inhibit acetylcholinesterase, because some individuals who have very little acetylcholine being released, or they have some other damage at the axon terminal or on the receiving nicotinic receptors, this will allow them to stay open longer, actually, to facilitate a action potential. Individuals who might be candidate for this type of medication might be individuals who have multiple sclerosis, or who have myasthenia gravis, and who may have had some botulism. Depolarization and propagation of sarcolemma. Once the motor end plate is depolarized at the neuromuscular junction, the depolarization will propagate across the sarcolemma in both directions. And how does it do this? It does it just the same way as it did it in the neuron. It'll open voltage-gated sodium channels and close voltage-gated potassium channels all along its way. And just like in a neuron, as soon as the action potential passes through a segment of the sarcolemma, that segment behind it will start its repolarization phase. The action potential will stimulate a muscle contraction. As that action potential moves across the sarcolemma, and it'll go down the T tubules that I talked about, it'll release the calcium stored in the sarcoplasmic reticulum. Then that calcium will bind with troponin, removing the tropomyosin, allowing the myosin heads to bind with the myosin sites on the actin and facilitate the sliding of myofilaments across each other, and that'll shorten the entire sarcomere, which will shorten the muscle fiber. As I mentioned before, action potentials are all or none event. Once initiated, it cannot be stopped. The entire length of the muscle fiber will contract. Just like neurons, muscle cells have a repolarization and a refractory period. The repolarization stage is going to restore the sarcolemma to its initial polarized state, to its resting membrane potential, actually. The refractory period is the stage or period of time where the muscle cell cannot be stimulated again until it's completely repolarized. Events at a muscle fiber contraction. Now we're inside the muscle cell. So in the relaxed state, myosin binding sites are covered up by that tropomyosin, and the tropomyosin is then held in place by the protein troponin. The myosin head is in the high energy upright cock position. It's ready to go. It's energized because it was energized by ATP phosphorylated, so it's bound to an ADP at this point. When an action potential sweeps along the sarcolemma and goes down the T tubules, it will cause the voltage-gated calcium channels on the sarcoplasmic reticulum to open, and the calcium will be released into the cytosol. Then the calcium will migrate to the troponin and bind with it, and that troponin will release the tropomyosin, exposing the binding sites on the actin filaments. Then the contraction begins. The myosin heads will bind with the actin, forming what's called a cross bridge, as you see in the example number 4. The myosin heads attach to the binding sites on the actin. Then ATP will quickly bind to the free site on the myosin head, causing it to detach from the actin. So what I want you to appreciate is that it takes energy to start a contraction and it takes energy to stop it. As long as there's ATP, that myosin head will continue to attach to subsequent binding sites on the actin, always pulling it towards the center of the sarcomere. ATP is hydrolyzed by ATPase into ADP, causing the myosin head to return to the cocked position, ready to form the next cross bridge to the next exposed binding site. This is almost a never-ending cycle and can continue to repeat as long as calcium is made available, exposing those binding sites, and as long as there's sufficient ATP. ATP is needed to start the contraction, or to start the cross bridge, if you will, to pull it towards the actin towards the center, and it's needed to disengage the myosin head. And disengagement is what's necessary for relaxation of the muscle fiber. This action of the myosin heads attaching to the actin myosin binding sites and sliding it towards the center of the sarcomere, again, is part of the sliding filament theory. This will result in the sarcomere shortening and therefore ultimately the whole muscle cell shortening. I want to remind you that the actin and myosin filaments themselves do not change length. It's just the sliding across that shortens the sarcomere. Well, as you might expect, if it takes a lot of steps to facilitate or initiate a muscle contraction, it's going to take a lot of steps to cause a muscle relaxation. An action potential can last from 1 to 5 milliseconds. It's on a timing cycle, so all this stuff happens automatically. So the acetylcholine esterase will start to break down the acetylcholine in the neuromuscular junction cleft that we talked about already. Calcium will be actively pumped back into the sarcoplasmic reticulum. And when you hear the word active pump, you know it's going to require ATP energy. This pumping of the calcium back into the sarcoplasmic reticulum will start about 1/20th of a second after the release of the calcium, so that's about 3 milliseconds later. The bond between the actin and myosin cross bridge will break, and that also requires ATP. The actin-myosin binding sites will be covered up again by tropomyosin and locked into place by troponin. Then potassium will diffuse out of the cell, and the sodium-potassium pump will restore the sarcolemma back to its resting membrane potential, and the contraction will end, and the muscle fiber will relax. The role of ATP in a muscle cell contraction is that it is needed to start a contraction and needed to stop a contraction. This becomes real evident after a person dies. Immediately their body goes into what's called rigor mortis, where their muscle cells are completely contracted, and that's a result from the lack of ATP, because they're not taking in any energy to disengage the myosin heads from the actin. So it stays in its cross bridge position until there's finally protein breakdown. We'll give you all the steps in order, starting with a nerve signal arriving at the neuromuscular junction and ending with the sodium-potassium pump, restoring the sarcolemma to resting membrane potential. Hypocalcemia. We already talked a little bit about this with nervous tissue. And here we see that it can increase the excitability of nerves tissue, but it also can do that with muscle tissue. In fact, hypocalcemia can cause mini end plate potentials at the neuromuscular junction, which really is not a normal finding. This causes small depolarizations that take place in the absence of neuron firing, and it moves the sarcolemma closer to threshold. That's why someone who has hypocalcemia can seem a little jittery. Types of skeletal muscle contractions. One is the isometric contraction, where the muscle does not change length and there's no movement of any joints, but the tension of the muscle increases. The example on the right shows a arm extended. It shows the shoulder joint in flexion and the elbow joint in extension. So imagine that this arm is extended out and has no weight in it. Then someone puts a 10-pound weight in the hand, and in order not to drop that weight, the bicep muscle is going to have to contract. But it doesn't change any length and there's no movement. But the tension in that muscle increases so that you don't drop the 10-pound weight. Now, the other type of movement or muscle contraction is a isotonic contraction. In isotonic contraction, the muscle does change length to produce movement, but the tension of the muscle remains constant. There's two types. There's eccentric contractions and concentric contractions. With the eccentric contraction, the muscle cell is extending. You see that on the bottom. So let me start with eccentric. So let's say we have that arm extended with the 10-pound weight on it, and you decide to do a bicep curl. That bicep curl is going to cause flexion of the elbow until the weight touches the shoulder. This is a concentric contraction. Think of it making a circle, creating a circle. Concentric is around, if you will. So the muscle itself is going to have to shorten in order to pull the forearm towards the shoulder. And so that eccentric contraction causes movement where the muscle cell shortens. Now you want to extend that elbow so you can do some curls. Well, that extension is going to cause the muscle to elongate, but it's going to remain tense, remain at same amount of tension so it doesn't drop the weight. And that is an eccentric contraction. What is a motor unit? Well, if you look at the diagram, you can see that it's showing some motor neurons exiting the ventral horn. Well, these motor neurons, it's only showing two. So one motor neuron is going to 3 muscle fibers, and the other motor neuron is going to 2 muscle fibers. So a motor unit is the one motor neuron and all the skeletal muscle fibers that are stimulated by that one motor neuron. You'll have a lot more than 2 motor neurons exiting a motor spinal nerve root, but this is just an example. And it's going to vary depending on the fine motor skills required of that muscle. For example, extraocular eye muscles will have very few muscle cell fibers per neuron, whereas the gastrocnemius muscle will have maybe several, could have 100 muscle fibers per one neuron. But the motor unit is what is the main unit that's going to stimulate a certain amount of muscle fibers to facilitate a contraction. Muscle recruitment. As the intensity of nervous stimulation increases, more and more motor units are activated. The strength or force of the whole muscle contraction depends on the number of fibers stimulated. More fibers contracting results in greater muscle tension and control. Muscles can continue to contract until they run out of energy. So I'm going to paraphrase some of what all of this says. Muscle recruitment means, okay, let's go back to the example where the arm is extended and has a 10-pound weight in it. Then say somebody else comes along and puts another 10 pounds on the hand. So now you're holding 20. Well, in order to do that, your brain had to send more signals to more motor units to contract more muscle fibers so that you would not drop the 2 weights. That's recruitment. Another example is, let's say you bent over to help a friend move some furniture, and you were anticipating in your mind, you mentally assessed about how much strength you would need to, say, pick up a dining table. Well, you go to pick up the end and you find that this dining table is made out of concrete, and so it's a little heavier than expected. And so as not to drop it on your foot or hurt your friend that's helping you move, your brain will send more signals to your whole muscles to contract more muscle fibers. Now, the other part of this is a muscle cannot contract without nervous stimuli. And without nervous stimuli, there's nothing's going to happen. So that is a key part in training your muscles, in exercise. You have to have a good connection to the muscle fibers. No stimulus, no contraction. So muscles respond to a stimulus. Here I've got all or none again. I guess I want to drill that into you. They're all or none. Now, another point that's important to wrap your head around is within a whole skeletal muscle, not all the fibers will be stimulated at the same time. If all the fibers were to be stimulated at the same time, that's actually a pathology. So you don't want that to happen. So intermittent different motor units, even if it's sustaining a contraction, will change combinations in order to let certain groups of fibers to rest and the others contract. So for example, if you were holding that 10-pound weight, and let's say you were going to hold it for 10 minutes, well, after a certain period of time, another group of muscle cells will contract and allow a certain other group of muscle cells to relax, so they can restore their creatine phosphate, they can restore their oxygen stores. And as long as you're breathing in enough oxygen and there's plenty of glucose and fatty acids to use to make ATP, you can sustain that muscle cell contraction until you have fatigue. Types of contractions. One's a twitch, a single brief contraction, a few fibers are stimulated, and this is not a normal function. I'm sure all of you have experienced once in a while you have a little twitch in your eye, and you know that's not normal. Something's going on, most likely upper cervical subluxation. It can be caused by stress, and it could just be an idiopathic cause. We don't know, but it's not normal. The muscles should not contract in the form of a twitch. Then you have what's called summation, where one contraction is immediately followed by another. Different groups of muscle fibers are being stimulated each time, and so the whole muscle does not completely return to the resting state. So it's really hard to tell when somebody's doing summation of which muscle groups are contracting and relaxing. Again, you're holding that 10-pound weight, and you're able to hold that weight because you have summation. The types of greater responses that are ideal for a long, strong contraction is the tetanus or tentonic contraction. This is actually the ideal type of contraction because it can hold the whole muscle in strength for such a long time that there's no evidence of whole muscle relaxation at all in between the different groups of muscle fibers, alternating between which are stimulated and which are relaxing. The frequency of the stimulation does not allow for any relaxation of the whole muscle between contractions. So this is just super summation. This results in a smooth, strong, sustained muscle contraction, which is the goal of weight training. You'll begin your weight training or at a summation of contractions where there might be just a tiny bit sensation of weakness or fatigue while you're holding a weight. But as you train the muscle, you can get it to the tentonic type of contraction where there's no fatigue and it can sustain the contraction for a long time. Because of this strong type of contraction, and it's called tentonic, that's how the condition, the disease tetanus, got its name from, because of its sustained contraction caused by the bacteria. In skeletal muscle cells, size does matter when it comes to strength. Skeletal muscle can generate about 50 pounds of force per square inch of muscle, or 3 to 4 kilograms per centimeter square of muscle. So if you take a small square inch of muscle, that square inch of muscle will have the same amount of strength in everyone who has muscle. However, some individuals will have more muscle cells than others, and that is mass. And that main difference is muscle mass and exercise. The strength of muscle is determined by its size or mass. Increased size equals increased strength. The more cross-sectional area a muscle has, the more muscle fibers it will have, the more strength it will have. Males equal females in terms of the strength of the muscle per cross-sectional area. However, a female athlete in the same competition class, for example, the same age, the same height, the same weight, the same type of training, the same type of nutrition, will have much less muscle mass. The main difference between males and female athletic performance with the muscle mass is due to testosterone versus estrogen. We'll learn in endocrine how testosterone is essential for producing more muscle mass. It stimulates protein synthesis and it targets skeletal muscle. Estrogen really does not do that much for muscle mass. It does stimulate protein synthesis, but it mostly targets the sex organs and not so much muscle mass. To increase muscle mass, strenuous exercise and resistance training and proper training will actually allow the body to produce more mitochondria in skeletal muscles, more myofibrils and myofilaments, and that will increase the muscle mass. You do not increase the number of muscle cells within a whole muscle. You're born with what you're going to get, but you can actually increase the size of the individual muscle cells. Resistance exercise is going to increase strength. It'll increase neurological recruitment, which is actually key because a muscle cannot contract without getting a nerve stimulus. It increases the number of actin and myosin filaments and increases the number of mitochondria. It'll also increase the myoglobin and glucose storage so that the muscle cell will have more endurance as well. What is the staircase effect? It is considered repetitive contractions will ultimately lead to a greater force of contraction. Not exactly sure on the science, but we think a lot has to do with calcium versus more protein in the body. That's because the calcium levels remaining in the cytosol after a contraction, which will be more available for subsequent contractions, and you know a contraction cannot occur without calcium. So repeated contractions at a high frequency will occur before enough of the calcium is pumped back into the sarcoplasmic reticulum, leaving more in the cytoplasm and increasing the strength of the contraction. Muscle tone needs to be maintained 24/7, and in the healthy adult it is. So even at rest, some muscle fibers are being contracted. This muscle tone is important or essential for maintaining posture and preventing you from falling out of your chair when you're listening to my lecture. It also prevents atrophy of muscles. It keeps muscles healthy. So what happens is throughout the day, different motor units are alternately contracting different groups of muscle fibers in every muscle of your body. So if somebody is developing atrophy, this system is not working. Stretching of muscle fibers. A muscle fiber can change lengths with stretch. It can even add sarcomeres to meet the length demands of the muscle. Now keep in mind, some individuals can stretch more than others. And a lot of that has to do with ligament laxity. So don't compare yourself with someone else if they can stretch a lot further than you can. But what you need to do is stretch your muscles to its maximum and allow for more sarcomeres, because the more sarcomeres you have in your muscle, the more sarcomeres will be able to contract and the stronger your muscle will be. Inactivity, though, can cause the opposite to happen. Lack of stretching can cause the body to remove sarcomeres, decreasing their fiber length. A good example I have of that is women in the 50s always wore stiletto heels. I can remember my mother actually wearing stiletto heels to do housework and vacuuming the house. She always had heels on. Well, as she became a senior citizen well into her 80s, the stiletto heels were no longer practical. But she could not wear flat heels any longer because her gastrocnemius actually shortened. And it took a long time of stretching, and she always had to wear these ugly orthopedic shoes that had a heel in it because her gastrocnemius shortened from constantly being in that contracted position. So please stretch your muscles. And I love this photo because anybody, any size, any age can stretch. Muscle fatigue. A tetanus muscle contraction can continue until the muscle fatigues. Muscles fatigue when the muscle relaxes even though stimulus continues. Have you ever gotten to the point where you're so physically tired, you keep telling your muscles to do something and they're just not listening to you? That often can happen with somebody, say they're running a marathon and they're close to the finish line. They know they may have only 3 or 4 miles to go, but they are so exhausted, even though they have the drive to go further, their muscles just will not respond. We'll talk about how muscle cell fatigues on the next slide. A lot of muscle fatigue is caused by substrate depletion. That means you've used up most of your glucose and glycogen stores and fatty acids that were available. So now you're just have a period of time where you've got lactic acid being built up because you're making ATP via anaerobic metabolism. And that can be caused from lack of oxygen, which I have on the third one. So lack of oxygen can cause that because you have demanded more ATP than the oxygen you're taking in. That's the most common reason of fatigue. And repeated stimulation, which can lead to depleted neurotransmitters. That doesn't happen too often. So the most common reason for muscle fatigue is lack of oxygen and substrate depletion. The lack of oxygen is quickly restored if an athlete at some point in their run will stop and just take in some oxygen and let their body's myoglobin get refilled in their skeletal muscle, and maybe stop at a station and get an energy drink, and then they can keep going. What happens when skeletal muscle is denervated? Well, it's going to cause atrophy. A muscle cannot contract without nerve stimulation. Atrophy can happen with subluxations if a neuralgia is significantly limiting nerve stimulation to certain muscle cells. Within about 2 months, degenerative changes will start to occur in the muscle fiber. If reinnervation occurs within 3 months, a full recovery can be made. After about 3 months, the total recovery becomes less and less, and eventually denervated muscle is replaced by fatty tissue. Kind of like I told you with muscular dystrophy. When a muscle is not functioning properly, it gets replaced with fatty tissue. And if it's not reinnervated by a normal nerve stimulation, after about 1 to 2 years, there's not going to be any function of that muscle cell. Now, I'm saying muscle cell, not whole muscle, so keep in mind that whole muscle is made up of thousands of muscle cells. So you see the image of the image of the leg that we see on the right is a normal size, and the atrophied muscle is on the left. You can see there's still some muscle function, some muscle there, but most of it has turned to fat cells. With denervated skeletal muscle cell, like you might see with botulism, the damaged axons can sprout new telodendria and form a new neuromuscular junction, and these will be called microunits. The microunits can contain as many as 5 times the muscle fibers for each motor neuron. So, as like in the case of botulism, the damaged cholinergic neurons can sprout new neuromuscular junctions, but the motor unit is going to innervate more muscle fibers per motor unit, so the contractions may not be as smooth or as strong. So that's why some of those individuals may present with residual muscle weakness. If a neuromuscular junction axon is severed as a result of an acute trauma, acetylcholine receptors, cholinergic, ionotropic receptors, nicotinic receptors, whatever you want to call them, they have so many AKAs, will spread out over the entire sarcolemma. This will increase the sensitivity, so any nearby neuromuscular junctions releasing acetylcholine, and that acetylcholine just dissipates out of the synaptic cleft. These other acetylcholine receptors along the sarcolemma may be able to bind with some of those and help that skeletal muscle maintain some contraction to maintain some health. This will increase the sensitivity of the muscle, and it's referred to as denervation supersensitivity. It's not going to really help that muscle for too long until that severed axon is regenerated, but it allows that muscle cell to maintain as much of a normal contraction for at least 3 to 4 weeks, which was the timing I've said on the other slide, that if nervous stimulation is restored, you can have full recovery. Muscular junction, and we did talk about those with neurotransmitters. And some of the drugs actually will destroy them, such as sarin gas, which will increase or sustain a muscle contraction because it inactivates the cholinesterase, and that'll allow acetylcholine to build up into the neuromuscular junction, holding those ligand-gated ion channels open, allowing sodium to continue to flood in. And it will, because the sodium, it doesn't affect the sodium-potassium pump, so that sodium-potassium pump is going to continuously pump sodium out, so sodium will want to continuously flood in, maintaining constant depolarization. The other drug is curiform or Curare drugs, and they block transmission of acetylcholine. So they don't even allow acetylcholine to bind with the ionotropic receptors. So the muscle cells at the neuromuscular junction cannot even contract, and you have paralysis. So the sarin causes severe contraction. You can't release the contraction. Curiforms doesn't allow a contraction.